I've been in a lot of conferences, and the worst time is actually giving a talk after lunch. And I'm coming from the Faculty of Mathematics and Information Sciences of Warsaw University of Technology. So bear with me. I'm not talking a lot about mathematics and statistics, which actually is my background. But I would like to share with you some reflections for you for the MECFS research community, mostly. So today, even at the title, I said that I will talk a little bit about long COVID. Mostly, I'm going to focus on MECFS, which actually what I've been doing in the last, let's say, 5-6 years. So a little bit of long COVID at the end, if I will have time, if Professor Vascarneiro allow me to share some slides on long COVID. But you can take some of my worries and reflections to the long COVID research field, okay? So, my talk is about research challenge on MECFS and long COVID, okay? And the title is In the Era of Machine Learning Artificial Intelligence. I don't know if you tried, but there is something called ChatGPT and other large language models. And what I've done, I'm already old school, right? I'm not, even though I'm coming from the Warsaw University of Technology, I'm still a mathematician and applied statistician, from the Technical University. So I was not so much, keen on using ChatGPT, but I give it a go for the sake of this presentation. So I asked ChatGPT, what are the MECFS research questions, okay? Think about this, and just for a moment, let's go. And I tried 4 large language models. The first one that you actually most people know is ChatGPT, then Gemini, then is Llama or Vicuna, and the last one is Copilot of Microsoft Bing, okay? And here are the main topics that the large language models actually provide, okay? And I'll just give you, just a couple of seconds for you guys to see it, which actually what is interesting is on top of that, on top of the, well, it's working? It does. Okay. But on the top, of course, the top research question is to know the causes, etiology, understanding the underlying mechanism. It was interesting for me that the last one, this Copilot of the Microsoft Bing, actually, it was mostly on policy side, which is funding challenges, diagnostic challenges, skepticism and stigma, attracting researchers, need for consistency, targeting. Okay, it's just they don't like me anymore, so they want to shut me down. Okay, so just bear with me, it's mostly is on causes, diagnostic, but I don't agree with this. And I want to provoke you a little bit on the discussion. I don't know what's going on. This is the problem of someone else is controlling your presentation. Okay. Okay, I think is this the screensaver at some point. Okay, for me this is the main question. Reproducibility. We need to have research that is reproducible. And no matter what questions we would like to answer, all of them relies on this premise, which actually you need to have research that actually being conducted that tackle all these things, but you want the results to be reproducible, because if they are not reproducible, you don't know anything, right? You ended up with more questions than answers. So this is the big elephant in the room. And we all need to think about that, because if you go to search the MECFS literature, you're going to see that a lot of things are inconsistent, contradictory, and our job is actually to understand why is that, we as researchers. And I just want to highlight this thing. This is not a problem of MECFS as a field, first of all. And actually, this is a problem that is being discussed in the science community. Sorry. And it's because it's not working. So you have, a very, opinion paper about scientific reproducibility. And if they ask you in a large sample of researchers, if there was a reproducibility crisis, and this was published in Nature, and it was mostly of 50% of the people said that there was reproducible crisis. And what does it mean is that people cannot reproduce the results that are published, okay? And so we have to be aware of that. So we have to be aware that every time that we read the paper, we have to take it with a grain of salt, and we need to see if the papers are, comparable in a way. If we are comparing apples with apples, orange with orange, or we are not comparing orange with apples, okay? So this is, and I bring you this case of rituximab just to illustrate the fact or the problem of reproducibility. This was a study. I would be very happy that Øystein Flug will be here. He's from Norway, and his team they start using rituximab in 2009 in a very small number of patients. So in ME/CFS patients, from the observation that cancer patients were better of fatigue every time that they was using this rituximab drug, okay? So they thought this was a very promising line of research, and they extended the study, and so they conducted a study in 2019, or they published a study in 2011, and it was a study on 30 patients in total. The results were promising. Then, 4 years later, in open label, I think, open label clinical trial, they also find, interesting results. And this was kind of like, that supported a larger clinical trial. And actually, when they extended the clinical trial to almost 150 patients, randomized to rituximab and placebo, the results were very disappointing, okay? And I think you have to have courage that put in the website, and this is actually in the Norwegian university website, that they say no effect of cancer drug treatment in patients with ME/CFS. Okay, after 10 years, with a very promising treatment, you ended up with a disappointing result, and it was really disappointing for everyone in the field. Okay. So I ask a question again to large language models, so that I try to convince you that it's not only me that you have this idea that reproducibility, we have like a problem. And I ask him if, considering low, middle, high, what is the level of reproducibility in ME/CFS research? And these are the answers. ChatGPT, low. Gemini, low. Vicuna, low to moderate. And low, because it depends on the large language models, because they include 2. And in the Microsoft Bing, the Copilot, they say it varies across studies. So it's an issue. I'm not giving you a negative, I don't want to give you a bad impression of this, but this is an issue. It's just we need to be aware what's going on. So what are the implications? And it's coming back to the presentation of Lyndon Tannenbaum, that this slows down progress. Reproducibility slows really down progress. And for us as researchers, we face this challenge. What line of research we should follow when we read the literature? Which one we should believe, okay? So obscure the most promising lines of research. Of course, for the society, the implication for society is that there is this belief in science, scientists, and for you guys, the patients, this is a problem as well, because then you realize, okay, ME/CFS doesn't exist, fibromyalgia doesn't exist, because the results are contradictory. You don't see any positive results, not consistent. So it must be something, in their minds. So it's this belief. It's a big, big problem. Of course, if you don't have reproducibility results, what the governments say, oh, we are losing our money, so we're not going to waste, our precious taxpayers' money in research that is not reproducible, so it in the end decreases funding. And for the young researchers panel, okay, this is a problem, because if you have a field that actually is not progressing, or you feel that there is this kind of blockage, people will try to find something that is much easier to do, trying to work for a company instead of doing research, and not at the end of a PhD, you're going to have negative results, okay? So let's go back to basics. It's really basic stuff that I'm going to discuss. Why there is a low reproducibility of studies? First, you have medical reasons and technical reasons, okay? And we have to tackle both of them when you do research. For the sake of this presentation, I invite you to go to the poster of my PhD student, João Malato. The poster is outside, and he talked a little bit about the technical reasons, which is due to misdiagnosis and other things like technical, statistics problems, not so thorough analysis. So talk to him about the technical problems of this reproducibility. What I'm going to focus on is actually on the biomedical reasons, and this is something that we all need to be aware of. So biomedical reasons is mostly about the biggest challenge in MECFS research, which is actually disease heterogeneity. And when I actually read papers, and I write papers, we all, all the research community, we write papers and we actually put these kind of like fancy words, but we have to be aware actually what does it mean? Really, what does it mean in practice? And this is what I'm going to bring you. Heterogeneity is composed of this, mostly on these 5 factors: symptoms, disease triggers, comorbidity, disease stage, and let's say pathophysiology. When I say pathophysiology, it is that you can have multiple pathophysiological mechanisms operating in the same individual, okay? So let's go to the symptoms. And I'm not advocating any diagnostic here, just as a disclaimer, but let's go just for the CDC criteria. Okay? You have unexplained fatigue for more than 6 months. We all agree with that, right? And then you have this additional condition, at least 4 out of 8 core symptoms, and then, beyond the exclusion of the disease. So you have all these 8 core symptoms, and you can pick up 4. So what does it mean in terms of statistical analysis? Okay? Don't worry. Everyone, I think you can go back to the high school, and this is the symbol of combinations. So in the end, you can have, if you sample a group of patients, you could have 163 combination of symptoms, okay, in a sample. And this is from the CDC criteria itself. It doesn't mean that you're going to observe all of them, but this is what is possible to obtain, okay? When you actually make a condition, which actually is used in CureMe, if I'm not wrong, you actually modify the CDC criteria and you say, I would like to have, beyond persistent fatigue, you're going to have a mandatory symptom post-exertional malaise, and now you relaxed the core symptoms, so now you have at least 3 symptoms out of 7, okay? And then in the end, you're going to have 99. It's not so bad. So you decrease the combination of symptoms. Okay? So now let's go to the disease triggers. And I think this is mostly, it's not new for all of, for most of you. So this is a study conducted, I think it was in the US, from the group of Montoya. And it's all epidemiology data, and they asked the patients to report what were supposedly the disease triggers that they claimed that caused the disease. And you can have, 5 factors, okay? The most prevalent is the infectious trigger, which is not surprising. We have been discussing that. And if you go to the infection triggers, here is the table 2 on your right-hand side. You have 6 categories related to infections. Okay, another complexity. If you go to the UK MECFS Biobank, and we did that, I did that work together with Ileana and Luis and others, and you can see that they also have 5 categories of disease triggers. Some of them are infections, some of them are exposure to toxins, something even in our cohort, you have even tropical infections. At least that's what people suggest that they cause the disease. So comorbidities, and this is something, probably I need to discuss with Luis. I'm coming back to our Euromean network, and we published a recommendation paper about the guidelines for diagnostic and treatment. And this is what we came about comorbidities. Actually, it's allowed all these comorbidities: fibromyalgia, restless leg syndrome, periodic limb disorders, POTS, and so on and so forth. You can read the list. So there are 7 combinations of these comorbidities. Actually, if you combine them, if you actually could take analysis of all these things, you ended up with that huge amount of comorbidities. Okay? What about disease stage? Disease stage is also something that we have to think about. And I'm just going to bring a paper published by Luis Nakula and Eliana, where they proposed that the ME/CFS natural history of the disease can be decomposed or divided into these disease stages. First stage is predisposition for the ME/CFS, then exactly the trigger for the disease, and then you have the prodromal and early disease and established disease. So where are you going to sample? Most of the things you're going to sample in established disease, okay? But if you take into account all this, you have, sorry, I'm coming back, you have 3 disease stages after onset. So you're already heading up the disease stage. What about pathophysiology? And I'm borrowing again from the same paper of Luis and Iliana, all the mechanisms that were highlighted in the paper. Okay? And you have oxidative stress, inflammatory response, central nervous system response, all of that. So what I want you guys to show is not the mechanisms themselves, is that you can have multiple mechanisms in the same people. And then it's a matter of combination of mechanisms that you're going to have. So in summary, you're going to see this. All these things are linked together. All this heterogeneity that when I was saying is about around 163, slash 99 combination of core symptoms, depending on the case definition. A lot of combinations due to comorbidities, 5 categories of disease triggers, 3 or more disease stages, depending on how you're going to define the natural history of the disease. And of course, and this is the biggest elephant in all these common things, actually you don't know the number of combination of these pathophysiological mechanisms, right? You don't know. You have to identify them first so that you can see how many combinations of these pathways can be involved. Okay, this is actually for long COVID, but I will focusing on that in the end. Otherwise, I won't have time. So I ask. So the question is that we are living in this era of machine learning and artificial intelligence, and the basic question that me as a mathematician and a biostatistician by training is exactly this: Can machine learning solve this combinatorial problem per se? Okay, and I don't know how you perceive machine learning, right? So I brought a very simple scheme of what it is, no mathematics, just the basic idea. So what you have is that you take samples, from patients, healthy controls, you put it, you collect it, and then you train several statistical models, computational models that try to differentiate between cases and controls. The quality of the predictions are okay. Then you can you can construct a final model that aggregates all these, let's say, these statistical models in a single model that can be used supposedly in a diagnostic or in a clinical setting. And at the end, you try to test the predictions with an independent data set. And then the hope is that you're going to have quality of the predictions very high in both training and testing. So, in theory, yes, machine learning can do that if there is a lot of data to learn from. And this comes again to the presentation of Linda Tannenbaum. We need to have collaborations, multicentric data that can feed these models so that we can tackle this combinatorial problem. So if you don't take anything from this presentation, is this. If we want to apply these machine learning algorithms that are very agnostic, we need to have good data to do it. Otherwise, we ended up with these kind of situations. Okay, I'm sorry. The problem of all this of machine learning is the generalization, which actually is in the testing part. So what you do is, mostly on the cases, is actually, you have, in the training set, you have a high quality of your predictions, which means like a study, and then you combine it in a final model, you feed the model with a new data set coming from a different cohort, but the cohort is actually smaller because you don't have access to a large cohorts. So the quality of these predictions are completely uncertain. You don't know what's actually happening. And this is mostly what happened in my paper, and I'm putting my finger on, I'm shooting myself in the foot, which is, we did machine learning exercise in a very nice data set published by the group of Carmen Schemberbogen and Charité. What we tried to do was use antibody response to Epstein-Barr virus, and with a lot of different antibodies, when we try to classify patients, distinguish patients from healthy controls. And we use machine learning algorithms. But the data set was very good here. On the right-hand side, it's just the number of antibodies in your classifier, and then accuracy of the predictions, if the predictions are correct, in a way. So you're going to have like the optimal number of antibodies that predict high quality of the predictions were around 27. But the problem and the catch of this paper is that in the validation was very high, but it was only 10 individuals. So when actually you do some calculation of the uncertainty on the testing data set, actually it's very big. So we had the hope that this is a good result, but we have to be fair and say we actually don't know because the uncertainty is very high, especially in the test data set, because it's only 10 individuals. You can do the same thing in the generalization. And this is a little bit what Jonas Bergqvist did. He's trying to do, you validate the model at a certain time point and the patients and controls, and then you follow the patients over time, and then your training, the testing is actually done on the same cohort. But because you heard about, Ileana and Jonas saying that the symptoms, they vary over time, is also expected that you're going to have probably bad quality of the predictions when actually you sample individuals in a different time point, because the symptoms, the biology will change over time as well. Depends on the symptoms. Okay, so this brings to we have to be smarter than that. So we have to not use brute force approach like this and try to be a little bit smarter than that. And the way to solve this problem, and I don't know how much time do I have, because I tend to, speak a lot. How to solve this combinatorial problem is actually identifying disease subtypes. And when I say disease subtypes, and actually this is actually in the large language models. I'll try to be as quickly as I can. This is actually one of the challenges highlighted in these large language models, identifying heterogeneity subtyping in 3 of these large language models. So they are right, but the context is wrong. You have to make it the case that the reproducibility is actually drives the reproducibility, the reproducibility or lack of it drives this problem of identifying subtypes. And basically what we want, and this is just to remember, for you guys to remember that I'm not the first one claiming all of this, okay? This is already being discussed in the literature since at least 2005 by Leonard Jason. And Jonathan Kerr also did some subtyping of MECFS patients using genetic data or gene expression data. For me, it's just conceptual model is that when you actually use this, you try to use subtyping, you have to mean a little bit what it means. So you have, in this case, is that for me, the conceptual model is that you have like a bunch of disease triggers, biological factors, and comorbidities. The combination of these will create a specific disease trajectory for a specific patient. And for that specific patient, you're going to actually try to do precision medicine. So you have to combine all these factors and then make the right decision. So in a way, what I'm trying to say is that the patients themselves, they have this conceptual model in their minds, because we cannot, I myself, no one knows me better than myself, a little bit like. So in a way, it's like you help, the doctor can help make the decisions, but at the same time, the patient needs to be empowered with knowledge of himself and herself, so that actually you can use this kind of approach, precision medicine, because the doctor is going to be difficult to do the precision medicine, because you have to go to the specificity of each individual. Okay? I'm just going to, in terms of statistics, you can try to cluster individuals. And when I did the UK MECFS Biobank, I tried to cluster the symptoms of the core symptoms, and we had 246 patients, and we had 41 observed combination of these core symptoms. When I use a statistical technique that I no need to talk about in detail, but it's called latent class analysis, you can actually identify two classes of patients based just on the core symptoms, just to reduce the problem. And in this specific case, class 2 were the most prevalent, around 80% of the patients, 20% of the other patients were from class 1, and the difference between the classes were mostly from tender glands and joint pain. Okay. Disease triggers, you also can do some time. 2 minutes? Oh, it's 2 minutes, 2 minutes, okay, it's good. Disease subtyping, again, so okay, it's okay. 15 categories of disease triggers. And now this is a way that you can just do like we did in several papers. We divide the patients in infection trigger, non-infection trigger, and non-trigger. If you have enough data, in the infection trigger, you can do viral, parasitic, bacterial. And if you have even more data, you can go to the specificity of the virus itself or the bacteria. This is so again, bringing more data into play. So the take-home message is this. We can do the problem of reproducibility, I think, can be solved by machine learning, agnostic approaches to statistical analysis, but that requires a lot of data. But it's on top of that, we need to trying to do a little bit precision medicine, trying to do subtyping of the individuals. And I think for me, the biggest challenge is actually to do that. Each patient is a patient, with his own specificity. You need to do some kind of subtyping. And so I tackle all these things from a little bit of these large language models. And I'll just acknowledge, first of all, to John for all the brilliant work for this conference, and for all my key collaborators, Francisco Westermeier, Carmen Schembenbogen, Ileana, and Luis. It's a pleasure that you brought me to this field. It's complex, but it's a challenge. And of course, my students, including João, and of course, my families.